5. Welcome, adventurers. I'm your guide, Sean McZoo. This is the Reptile Reporter Show. Time to start the music. All right. I introduced myself. This is my partner in crime, I guess we should call him, Bill Strand, or the evil emperor who keeps convincing me to do things that I don't have time for, but sound like a good plan.
I thought I was the hostage.
Oh, the hostage. All right, yes. The headmaster of the Chameleon Academy, Bill Strand. All right, there we go. And Michael Nash. Why don't you introduce yourself, Michael?
Hey, how's it going? Good to be here. Thanks for helping me take a break from studying. Appreciate it.
Yes. So, yes, I can tell you what, that test, and we won't talk about it, but it's not too tough. It's only been 30 years since I took it, or 27 years since I took it. So I don't even know when we took taking in it, but you'll do perfect. So, all right. So first, Bill, announcements. Any announcements?
Well, only that I just released the Chameleon Academy Journal. Very proud of it. It's a passion project of mine. Really enjoy this new type of outreach, and it's really meant for those of us who want to take a break from the hustle and bustle and distractions of the world and just sit with a nice cup of coffee and flip through a Chameleon magazine. And
wait a minute, we're going to flip through it? I thought it was online, Bill.
It is, and you're going to digitally flip through it.
Oh, I don't even know if I can understand that. I'm a child of the 60s.
Yeah, get with the times. I'm going to have to send you a hard copy every 2 months.
Send me a hard copy so I can sit there and go through it and, drink my coffee, which I don't drink, and go through it. So that's okay. All right. Anything else, Bill?
Yeah. As a celebration of me releasing my first issue, my brother gave me a Lego Explorer with a little chameleon.
See, I told you this adventures would begin, and here we are.
That's right.
Hopefully everyone's buckled up for how much excitement we're going to have tonight. So don't let yourself fall out of the seat, because
I don't know. How are you going to beat this?
Yeah, I don't know. I don't know. But I heard that Michael's got some great information to throw at us, and we're going to draw it out of him and what have you. But all right, so for me, podcast is still up. It's been 3 episodes. Episode 4 launches on Tuesday. It's Two Turtle Tom. I'll tell you and me discussing all kinds of things, including how Bill got us into all this stuff. So, Bill's Tuesday Night Live. This is Wednesday Night Live. Thursday night is Two Turtle Tom. This all fell into place because of Bill. So he's the guy pulling all those strings in the background, kind of like the evil emperor from Star Wars. But we hope he doesn't have us going to war with each other. So we'll figure that out as time goes on. Social media, I've decided, with consultation with the emperor himself, that headmaster. Headmaster. That I'm going to start working more on Instagram than on Facebook. So I'm going to see what we could build over there. I took a picture of my Facebook status or my Instagram status on Reptile Show Reporter, and I put it into my pictures of social media stuff, so I know exactly where we are today. In a month, I'm going to take another picture and see where I'm at. And if it's positive and great, I'm going to, check the chat. All right, Bill. All right. I told him I got it written on the top of my page, but I'm not doing it. All right, chat. Here we go.
It's my job to remind him to switch to the chat. He's on the control.
Twitter Tom. I'm still not used to seeing my photo in the intro. Well, we can cut that. All right, Mikey Ben. Hello, everyone. Two Turtle Tom said hello to Mikey, so we'll let him go. Eliza Ann, you made it. So Mikey said hello to Tom. All right, this chat is going on without us.
Yeah, that's right.
I'm extremely boring tonight, Bill, because people are talking without me, and they're so all right. So Facebook user, way to go, Bill. I don't know who that Facebook user are, because I got a caption for that. Let's see, Facebook users, please be sure to include your name in every comment so we know who you are.
Unless you want to be anonymous.
Yes, unless you
I
want to
take
be
encouragement from anonymous people
That's
too.
a very famous author because I see that all the time. Anonymous, anonymous, you know.
You don't have to share your personal information to encourage me.
Okay, there we go. Plus, we also get to know who you were when we follow it up and we look on the replay because it does actually say who you are. All right, so let's see here. Chat. Let's go back to the chat. All right.
Joanne, hey.
All right.
Hello. We know Joanne.
All right. And at Two Turtle Time, Eliza Ann found him. So we've got a lot of stuff going on in the background. Nothing to do with the show or anything about it. Poor Michael, he was supposed to be studying for a very important test that's going to depend on the rest of his life. And here he is talking to us as we're chatting away. All right. I need to get rid of this Facebook user.
By the way, we should tell everybody, what is he studying for?
Oh, I don't know. Does he want anyone to know or not?
Hey, Chris.
Yeah, I can say it, I suppose, but it's called the Step 2 exam for medical school. You need to do it to demonstrate some basic like clinical proficiency, but it's kind of an interesting test because it's not truly pass fail. They care about how well you do on it. So it's sort of like if you take a driver's license test and you don't do that well, you can drive, but you can only drive maybe a Mini Cooper. Right.
Well, let's hope you get better than a Mini Cooper. I don't know. Is that I'm going to get myself in trouble. I'm not going to talk about what kind of patience that would be. All right. So since I took the test 30 years ago, I can't help you. I did pass, or at least they let me work. I don't know what am I don't remember the score, but they let me work, and I've been doing it for almost 30 years. So that was what 96 that I took that test. So depending on what year you took it, but all right. So hello everyone. Zev Green Reptiles. Who is that, Bill? Do you remember?
That's Chris.
That's Chris. All right.
Oh.
Hi, Chris. Quality of work is what they look for. All right, Andrew. All right. So let's get into it. Michael, what are you breeding right now? I've seen all these cool things. I saw that you just were on Facebook again. You were on the Reptile Enthusiast or the Chameleon Enthusiast, and you had this awesome orange chameleon. What the heck is that?
Yeah, I'm breeding most of what I've been working with for a while. I have a few new projects that I will wait to describe when they're a little further along. But that orange chameleon was a first for minor that I hatched out of an egg maybe, about a year ago now. So I sent off some first for minors, but I kept some obviously to keep the breeding going. And that's the most orange male I've ever come across. And he's had those colorations for 5 months now. So it doesn't seem super transient. He might actually stay with it. I hope so.
Wow, Bill, have you come across orange first for minors or?
I had a male that was very, very orange. He eventually went into his brownish colors, but that doesn't mean yours not going to stay orange. So
cool.
we're hoping for the orange to stay.
That's cool. So what else do you have?
Yeah, so something else I include there. I have some Burkesia stumphy that just hatched. I've had a lot of hatches, but recently I had some new ones come out, and they're looking rather orange as well. The sire, I don't know that this tracks at all, but the sire is very orange, and he's been that way his whole life. So I'm thinking that will stick. Have some Burkesia Abinawi that just hatched. That's exciting. They're very small. I have some Bragui pairing up now, some Thili, however you like to say it. Lots of good stuff going on. Small Kalumas with Phalix and Roloco and Lanatum, all those good ones. Some Christophers from my friend Chris, who's a really good guy. They've been.
Where do you keep all these things? I didn't have that big a apartment when I was in medical school. Okay, I had saltwater fish, but still, and iguanas and what have you. But nonetheless, I still didn't have that big of an apartment. Where do you? These are all small chameleons, I guess. That's probably where how you're keeping all of them. But that's why you're small. Okay.
Yeah, I mean, right now I have a lot of them outside as well. There's some challenges with the Colorado climate. Of course, it's very dry and somewhat hot, but it's amazing what they thrive in and do well with, as long as you can like keep them humid and provide everything they need in shade. But that said, actually, Furcifer antimena is another one that does great in climates like this outside. I have 3 different bloodlines incubating there. I hope they hatch pretty soon.
That's awesome.
I love those. Yeah, Furcifer antimena, I think, has a lot of potential. I say that about everything, but
we know, but we're just going to let you keep saying it because you have such cool things. You know, that's great. So
yeah, some lateralis about to hatch soon. Some peteri, I have 3. Well, I guess 4, but a couple small clutches. At first for peteri that I'm hoping will hatch pretty soon. Yeah, some Wilsii. Lots of little.
So what's the total on the number of species that you have?
You never ask a man his number.
It's in double digits, but maybe that's not a surprise.
Yeah, well, I think you just rattled off a dozen, but okay. But I wasn't counting, so okay, all right. I'm not going to ask a man his number of species. I'd hate for somebody to ask me how many species I had or how many chameleons in each species. So, and how many actual species I have in the house. You know, there's us, there's the humans, and there's the puppies, and yeah, there's a bunch of other stuff here. If anyone's asking, right?
I make it a point not to know.
Exactly. All right.
And that's the safest.
Right. Exactly. Okay. All right. So that's what you're up to. What is this newest, latest kind of thing that's in husbandry that we're talking about that you mentioned? There's a paper on that talks about telomeres and all that kind of stuff. Yeah. What is that?
So, I mean, there's a lot of elements of husbandry I think are important to discuss, but, you know, this paper came out in 2020, and this was with a bunch of field scientists and other scientists studying Furcifer labordi and Furcifer nicosii. So 2 different species that occupy somewhat similar habitats. And basically, the final conclusion of the paper was that in response to environmental stressors, the Furcifer labordi had their telomeres shrink and basically had a programmed self-destruct almost, where they died because their cells decided to stop working effectively. It's correlational. So, you know, we don't know exactly every specific about what's going on, but the point is it's quite interesting because it suggests that the environmental stressors have a defined molecular role in determining the lifespan of these animals. And a follow-up study in another journal also found that depending on the duration of time the wet season happens for, you can get extension of the lifespan of Furcifer labordi. And for context, Furcifer labordi, little chameleon, that it has the shortest lifespan of any tetrapod, any land vertebrate. So that's why they're looking at it. Why does it live so short? But what's very interesting is it suggests that overall, what we do in captivity might have this clear effect on the dynamics of every cell, right, in the animal. And we kind of knew that, but now we have a better way to quantify it. We have a better understanding of what's really going on, right? So that's important because we've been talking more and more recently about simulating a dry season, right? And simulating a wet season and seeing if that's going to improve longevity. But what I'm thinking is we're kind of in a potentially dangerous spot there, because if we simulate too much of a dry season in certain species, we might get this telomeric shortening, and you can't really come back from that.
So let's talk about this whole telomere thing.
Yeah.
It's related to DNA, which probably most of our viewers don't know about, and it's as people age, the telomeres get shorter and they don't fix, and that's how we think of aging. That's, I think, what I've read of recent. This is new stuff since I graduated many moons ago. So what is the telomere? What is it part of, and how does it work?
Yeah, I should have probably led with just kind of a basic idea of what I'm even talking about here, because the telomere is basically a region of the DNA or chromosome, whichever way you look at it, that's on the ends, right? And it's there specifically as a buffer to protect all of the good, useful DNA between the telomeres. So it's sort of like protection for the DNA. Every time a cell divides, its DNA gets a tiny bit shorter each time. And that has a big role in determining how long you're going to live. So the length of the telomere is basically how many divisions are proportional to how many divisions you can get through before you start losing DNA that matters. Right? So the longer the telomere, in theory, the longer you could live. That's not ultimately how it works, but that's the theory, right?
There's a lot out there about how to get yourself to be younger, how to live longer, how to make your telomeres longer, how do you age a person, how do you do your biological age as opposed to your chronological age. We talk about, you know, in humans, we talk about the well-being and their health span as well as their lifespan, and the telomere age is kind of that health span, and the lifespan is, you know, what have you, so in chameleons, is there any way to get them longer? Is there anything that, I mean, I know that there's some stuff in humans that may make it a little bit longer, may repair the DNA a little bit, but but the problem with making that work is then you can cause a cancer, right? I mean, if you get the DNA to start going crazy and it proliferates and gets bigger, you might cause a mistake and then it's cancerous. And if the immune system doesn't wipe it out before it wipes you out, then you're shorter lived as well. So is that correct?
Yes.
This is all, you know, this is kind of what I've, you know, I did have to look this up.
Well, this is a fine mess you've gotten us into.
So I did look this up last night to make sure I could even talk intelligently about this, if that's what it seems like. But okay, so that's where we're at. So we're figuring out maybe if we put this dry season with this chameleon, it doesn't live that long, we're going to kill it off because its DNA is going to be corrupted. It's going to wipe it out. And it's so all that stuff Bill was saying about a dry season, we shouldn't be doing that. Is that the conclusion? I'm sorry.
Yeah, I think doing black and white things here is not gonna get us far.
So I wouldn't say that's my conclusion. Right?
Okay, it's my conclusion, but it's errant and it's erroneous, but it's still a good conclusion to come to because.
Well, that just kind of cuts my year short.
All right. I told you I was going to be
nothing more to talk about.
Bill was supposed to just smile and wave, and Michael was going to do all the stuff. So that's what we're doing here. That's our roles.
It's kind of interesting, right? Because you certainly don't want to trigger. And again, I need to say, like, I'm not claiming that this is true across all species. I'm not claiming that this is like what happens for sure, right? But I think it's important to discuss these ideas as they come out, and we have some evidence that this is what is going on. So more just for discussion here. But the thing is that if you look at Madagascar chameleons, and many chameleons, they're super seasonal creatures, right? I mean, just the fact that there's species that are annual and they die every year, all of them. We know that it's not just some tropical paradise where it's perfect 75 all year round. Even areas like the rainforest, there's changes in precipitation rates, there's changes in humidity, like there's different stuff going on environmentally. So for a lot of the less common species, it seems that reproducing that dry and wet season is essential to getting them to breed. And I found that with a lot of species. But what I'm saying is, if someone is specifically trying to keep the animal as a pet, right, it might be that we just need to modify how they're kept versus people trying to breed them, because it's a balancing act, right? So I wonder if in the future, as we learn more about this, it might actually lead to a pet care sheet, for instance, versus a reproduction care sheet.
Interesting.
As a split.
So you're creating more work for Bill because he's worked so hard.
Apparently.
He's not even got care sheets. He's got care booklets now. They're like They're like
Yes.
so long for each different species. They're like 65 pages. You have to listen to them on Audible because you can't even page through them. They don't even exist in paper format anymore. But nonetheless, he's now going to have 2 of them, one for pets and one for breeding. All right, so that's good.
So much for retirement.
Yeah.
You can retire, Bill, when your telomeres get short enough. So we're measuring, and you're like 35 via your telomeres, so you got to keep going.
Great.
Yeah, so it's just kind of an interesting idea, right? Because in theory, for instance, this year I had some firsts for Wilsii outside a few months ago, and it was dry. It was dry out. Ambient humidity was like 15%, right? And what I found is with Wilsii, they actually dug and they would hide themselves in the substrate under kind of debris, right? And that's not unique to just Wilsii. You see that in a lot of different chameleons. So what I'm thinking is you can trigger a dry season, sure, but that might be at the expense of the long-term animal, lifespan if you go too far with it. So I think ultimately what might work, again, we need to do more to figure it out, is for pets, you keep them right at the threshold between dry and wet. So they don't have this need to reproduce, and yet everything else is toned down a little bit. So perhaps that would allow them to live longer. They found actually right after reproduction, Furcifer labordi telomeres, they shortened. Just the act of mating, it just cut the lifespan short just like that. So, and that kind of makes sense. You breed, you're sort of done, you're stressed out, whatever. You're going to be dead soon. Let's just make it happen.
Wait a minute. That's not true in humans because you got to wait at least 18 years and now 25.
Oh, yeah. Now Sean wakes up.
So they make it so they can leave the nest, and the nest needs to stay there for 25, 30 years. I don't know. I've got to 25 with my kids. And so 25 and 23 have left the nest, but I'm not sure it's permanent yet. So we'll see. Anyways, I digress.
Yeah. I mean, of course, we have a totally different strategy, right, of living than chameleons do. So it's all about just thinking, well, why does this exist this way? And I mean, it makes sense. Like, well, if they've evolved over all this time to always just be dead, it would make sense that the telomeres would respond accordingly. So, I do the seasonal fluctuation, and frankly, I think that's been really, really important as far as allowing breeding for so many of these species. But as I send more out as pets, they're captive bred, I'm starting to think, well, ultimately, it might be better to not even get them into those extremes. But we can still modify the parameters to keep them in that state where they don't have extremes on either end of, I need to be super active, I need to breed, or, oh, I'm almost going to die. And we all knew that already. But now, again, we have molecular reasons why that might be what's going on, which is exciting.
Yeah, and I'll say this is all part of exploration. Whenever traditionally we've done and I'm talking from the guy who makes care guides, and so I really think about all of this all the time, because when you put together a care guide, you gotta. And we've been doing 365 days of ideal conditions. That's generally the care guide. You get one care guide. Doesn't change throughout the year. And so this is just stepping into this area. Whenever we start looking into something different, it's going to be this wild frontier where you've got these advantages. Oh, okay, we'll give it the dry season, and then they're going to cool down and they're going to rest, and so they're going to extend their life. But now we're getting these. Okay, but wait a minute, some other factors may come into play that may shorten their life, and so that's the new frontier, is figuring out, okay, what of the dry season do we want to incorporate? And what do we not want to incorporate? Maybe we can make a dry season happen with the benefits if we only make it cooler and not drier. And who knows? I just pulled that out of the air. But this is us exploring, and who knows what we're going to end up with. We may end up with, it's better to have 365 days of the ideal tear tip, and then my care guides go down to 2 pages.
I'm not sure, Bill. I mean, like when you talk about humans, which is what we know a lot more about, no stress is not the best way to go. A little bit of stress, because with no stress, you just start to wear out, and your body doesn't fix itself. It doesn't do anything. Our mitochondria, which are the powerhouse of the cell, don't get stressed. They don't reproduce. But at the same token, if you take stress to the nth degree and you do a marathon without exercising beforehand or whatever, the first guy who ran the first marathon didn't live. He died after it. So you know, there's
he had a pretty intense training regimen before he ran his first marathon.
But at the same token, cold therapies, they're talking about hot therapies and different things that humans are talking about. I wonder if there's a little bit of a wiggle room where you're not having a perfect day every day. Do you stress a little bit? And I guess the question is, where do you draw the line? Because, people could run a couple miles every day and everything would be great. But then they go and they run a marathon and they tear their body apart and, so have you. But what is the chameleon type middle ground? And I guess we haven't even thought about it. We're just like, I need to set up this chameleon perfectly so that it has a perfect day every day. But I guess the question is that good?
Well, you can see how I've tried to gingerly step into that with my care guides. And you'll notice when I have the wet season and a dry season, the dry season is the hydration that we know works. They're going to be very well hydrated. And with the wet season, I add more to it. So it's like, okay, now you got more water than you need. So when I say dry season, that's really not the dry season. It's just a drier season and a little bit cooler season, not even getting anywhere near the stress limits. And so we're just gingerly taking baby steps towards exploration here.
Yeah. That's really good point, Sean, as well, about, some stress is necessary, of course. And another kind of example of that with some chameleons is Verkesia decarii, which should never be imported, but they get brought in by accident. I've bred them a number of times, and the best way to get them to breed is to make the environment really harsh. That's the only way I've been able to trigger breeding. It's very hot, it's dry, and they will go under the leaves and stop moving, stop eating, stop doing anything for a few months. And then you make it really hot, really humid, lots of rainfall, and then they all come up and breed. And
wait. You said hot and dry? Did you mean cool and dry?
Sorry. Yeah. Cool and dry at the beginning. Okay. Yeah. To trigger that dormancy state.
Okay.
Yeah. I misspoke. And then hot and lots of rainfall. And that's pretty extreme. So extreme that they go and hide like in the ground. And that's reproducible behavior that you see. And I think a couple other people have tried that with them, and that's essential. So, our idea, right, of what's uncomfortable might be very different than theirs. I'm not suggesting everyone, blow dry their chameleons, obviously, but it's.
Oh, well, we need to like put a
well, I guess say we got to
wording on that. Don't blow dry your chameleons ever at any time for any reason.
We also have to take into account things that we've been touching on here, that nature doesn't care about you once you've reproduced. And when I've had Mark Schertz on talking about the reproductive strategies of some of the Kaluma, the very small Kaluma, and how the hemipene structure is actually meant to damage the female on the way out to prevent insemination from other males. I mean, these chameleons, just the act of reproducing is messing them up. So maybe we just can't reproduce them if we want to go for the longevity. I mean, that would make sense, but we think about it as stress on the female body, not on damage, which is happening in nature.
Right. And you even see that with Labordi. You see them die. I've never been to Madagascar. I'm going to go this year, but I've never been, but from everything I've read and observed, they will die. Males will die just from extreme cortisol, or the equivalent in reptile, release from fighting so hard. And that's okay evolutionarily. Like, they've decided it's okay if we die fighting because enough will go on to make the new generation, right? That's built in. So, I mean, it's incredible. Our judgment as far as what's too much or not too much, it just doesn't always line up with what's going on in the wild. And that's part of the fun of this, I think, is trying to make it line up, which is hard.
Yeah, well, for us to make an intelligent decision, we actually have to know what all the parameters are. We bring in all these parameters and figure them out. Have to sort them out.
Right. Yeah.
Wow, I'm having a hard time not keeping up with what the conversation here and the conversation in the chat is good. They're going at it different from all about the same thing, but just the perspectives of, what if we have a pet? What if we don't? So, but yes, this is all very interesting. And the question is, how do we even figure this out? I mean, is there a DNA test that we could check? I mean, obviously they were able to check the telomeres of the chameleons. Is that something that's commercially available? Was that something that they just did themselves by looking under the microscope? I don't even know how you do that. What is the process to do that?
Yeah, so there's surrogate markers for telomere length, and then there's actually like measuring it. So you can look at the blood to determine like the ratio of different types of white blood cells. And that can give you kind of a proxy for likely what the telomeres are doing. So you just need blood, or you do a PCR, a polymerase chain reaction, and then you basically sequence just the telomeres. And based on how much DNA you get back when you amplify it, like you grow the DNA, that tells you how long the telomeres are. So really, you don't need much sample, and it's a simple test.
But these are tiny little chameleons, right? A small sample is a lot of their blood. So okay.
Right.
All right. So I'm just trying to put a for real perspective on this, as we're trying to get a chameleon's vein, and to get the blood or artery, I guess, if you need to get it that way. I don't even know how you get blood from a chameleon, but
the tail.
Injury.
Yeah, there's not a whole lot of blood in there to get.
Right. So okay. So all right. So where is this going to go? What's going to happen? How are we going to find this information? Where is it heading? How does that make, I mean, I know this is science that's way, way before we start to do anything with it, but what's the expectation? How long is it going to take to give us information? Are other people working on this? Is there other stuff going on? I didn't actually read the actual study. So is there future suggestions for future research? Is that actually going to happen? And who's doing all this?
Yeah, I mean, so that's the thing. The papers I found in First for Labordi, there's a lot of them because it's kind of an interesting species, right? The shortest living tetrapod. But a lot of the papers I've seen, that's not like the focus of the researchers. They don't just study First for Labordi all day, right? Some of them do. But it's sort of just, look at this interesting thing we could do by applying our science and our tools to this interesting question in a chameleon, right? So, there's a couple ways forward. I think people are slowly looking more into this, but as far as relevancy to keepers, that's the big gap, right? So I won't get too much into the specifics. I've been involved with a new kind of research project where we're doing some sequencing of some chameleons just to figure out, like, for instance, Kalumas, what's their method of sex determination. So we don't know these basic questions or the answers, these very basic things, right? So there's tools out there. There's some interest, but bridging that gap between data and what do we do in captivity, that's tough. What I'd like to do in the future whether it's collaborating with people or just on my own, I want to get a baseline telomere reading on various species that are imported regularly, just as sort of a general idea. I'll standardize it, of course, as best as I can, to see, well, based on that, can we determine the suitability of a species for captivity? Right? If they're just short from the beginning, which is the case for something like labordi, it might be that we can focus our efforts more appropriately if we just see, look, this creature, it's not going to live for a long time. Right now, we guess that based on natural history and just experience of people. But it'd be good to have some baseline understanding what types of chameleons are going to be good to have in captivity. So that's something I'm exploring myself. I hope other people pick it up. But
so is your hypothesis then that those that have the longer telomeres are going to live longer, and a longer lived chameleon is a better one to be in captivity? Is that what you're getting at?
Yeah. And, you know, there's to any telomere biologists that might be listening, or I'm aware there's during different stages of development, there's different levels of telomere.
This is a low level conversation. We're not talking about specifics or definites. Our audience is probably like looking through books right now as they're trying to figure out what's going on, except for Turtle Tom, who he deals with tortoises and turtles, which live 50 to 100 to 200 or whatever years. Their telomeres are probably 200 miles. So, or their cells reproduce so slowly that they never really chop their telomeres. I don't know what it is, but somehow, you know, the tortoise versus the hare. But those tortoises can go awful fast some days, though. You let one go loose in your backyard, see how fast it goes across. But nonetheless, they seem to be a lot slower than chameleons in general. So, but yeah, so we don't have to talk about specifics. We're not going to hold you to any of this information. This is just for curiosity's sake and for all of us to chat about tonight. So, yeah. All right. What else can you tell us?
Well, I want to make one point really clear. There's a lot of scam artists out there who sell things like telomerase cream and things like that are supposed to lengthen your telomere. Like there's no evidence behind that.
Oh, man, I'm going to have to return my entire bottle of telomerase cream. I got a bucket to dive in tonight. All right, okay.
I just want to put that out.
Cancel my subscription, Sean. I've had it with this.
I was about to start selling that. All right, okay. I'm going to have to cut this out and delete this after it's done. All right, I'm just kidding. No, but, you know, people will try to do anything. You know, as long as it doesn't hurt them, you know, why not try? But yeah, that's okay. So I'm sorry.
Let's have to get that out of the way. There's so many products that prey on that. But one other, it's a little bit complex. Well, I'll try to keep it at least somewhat tolerable. Another aspect of all this is that depending on the life stage of the animal, you have either rapidly decreasing telomere or slow, right? You can actually change that depending on if it's a baby, if it's older, and if it's a long telomere across various species at the beginning, like for the animal, right? It's the longest it's been. The period of shortening is fastest. Once it gets cut a little bit, it slows down, right? So there's this other element of, well, in a certain stage of growth, like juvenile stage, is that the time where the longevity is determined, right? If it's naturally getting cut faster or shorter, if we prolong that time at a certain developmental time point, that might lead to more telomere to burn later. So the lifespan might be longer. So we have to think about, well, what stage do we want to apply stress, reduce stress, because it's a dynamic process, and we could maybe, by treating them very well at these early stages, we could be adding years on their life later. So that's also some,
or by treating them, I guess, better than well, but just treating them properly. because it might be a little bit of stress that you apply at that time that if you apply no stress, it goes the other way. I mean, we don't know all that stuff. I think that stuff is still up in the air, but if you do it the right way, if you make the right decisions at that particular time, maybe it will lengthen their longevity, or maybe you'll create a crash at some point. So,
yeah, build chameleons, you treat them extra special and they will get unhealthy and produce 100 eggs.
Yes. But 100 eggs is good, Bill, because you have 100 little individually raised chameleon eggs, and I'm sure you have 100 cages, and you know, they've did their duty. They've reproduced, so they don't need to be there anymore, right?
Right.
Hey, where they come from, do they survive more than a year in their natural environment? No, none of them survive, right?
Well, this is an interesting question. Right now, that may be the case. But there has been discussion about whether that was always the case, because are the trees there, which lose all their leaves, were those the original trees before humans came in and messed things up? And are these introduced? So there is question as to whether they always did die at the end. But once again, we're learning a lot as long as we are willing to explore.
And there's always so much to learn. We could live 100 lifetimes and not really ever learn it all. But and every lifetime it gets more and more. So it's we'll have to see what to do. But okay, all right, Bill. Any final questions for Michael?
Oh no, I got a lot to say.
All right, you've actually stumped Bill. I'm going to have to give you the stump Bill.
No, this always happens when I bring Michael on.
The Make Bill Quiet award. I don't know if I've ever given that. I think, Michael, you've got that one. So even if you don't pass your test, I'm going to write to the people in charge of you to tell you that you're brilliant and they should just pass you anyway, since it's not a pass-fail test. All right.
Yeah, we know that none of these questions are easy, and trying to figure out an entire season and what's dangerous and what's helpful, and realizing the dry season is different for every species. Yeah, it's incredibly harsh with labordi that just get up and die and say, forget it, we're done. But you go off for pardalis, and you go during the dry season, you're going to see them hanging around and just saying hello. And so it's different for every species. And so this is a hugely complicated question.
Well, that's why I'm thinking, if possible, like to figure out the telomere stuff, it's not hard. It takes like a few hours. You just need expensive equipment. So the samples I have no problem getting, and lots of other people don't either, but it's just the equipment. So that's what I'm saying is not just based on what we observe, like, oh, well, it looks like pardalis does stick around. We know that, right?
Yeah.
But if we could have some objective metric that's easy to obtain, like telomere length, potentially, it'd be nice to be able to stratify each species by, like, what's the molecular reason this animal lives a certain time. And that's exciting for humans, for everything, right? So in the future, I hope somebody, or maybe me, establishes some objective way we can decide what is the likely lifespan and what do we need to do. That's sci-fi stuff, but maybe someday.
No, it isn't. It's, if you don't dream it, you can never get it. So that's we look forward to good things from you, Michael. Not only all these new chameleons that you're going to be able to send out, but also all this extra information. So, all right. One last question. How's it going with the Burkettia? Can you actually talk to this, or?
Yeah. I mean, I had some stumpy hats just yesterday. I've sent out some Burkettia. I think, maybe as soon as next month, I might be able to send out some captive bred individuals to, I'll say, the public. And that's because the ones I've produced thus far, and, that's like Feli and Stumpy and a number of others, the ones I produced that have gone out of my facility have gone to zoos. They've gone to conservation organizations. They've gone to like the Atlanta Botanical Garden, right? So right now I'm trying to send them off for more or less conservation purposes, but I'm about to get to the point where I can send more off as pets. I've sent some to some close friends of mine as well who keep chameleons. So I'm working on it this year. I think at least Stumpy and Feli. I should be able to sell more to the public.
That's awesome.
Well, yeah.
That's awesome. Okay. Bill, any final thoughts now?
No, I'm done.
All right.
Having fun. We'll continue to explore.
All right, Michael, any final thoughts before we get going?
No, yeah, I know that this is maybe one of the less accessible topics just because it's complicated and I'm omitting a lot of extra, believe it or not. But I think it's exciting to think about. And I'm glad there's any audience to push things forward and consider all this. I think it will be better for processing this stuff and looking to improve how we do things.
All right. Well, I wish you the best on this test coming up. I hope this was a good break for you to relax, and I hope we didn't torture you too much about all this stuff. It's probably low-level stuff for you, but I would warn you, or not warn you, I would advise you to trust your gut. That's always what they always told me. Trust your gut, because something inside of you tells you the right answer. So trust your gut. Anyways, everyone, thanks for being here tonight. We appreciate it. We'll be here next week at same time.